All right, in this chapter, we're going to talk about DNA and RNA structure and function, and then we'll go into a little bit about biotechnology. So we're going to start off with an overview of DNA and RNA structure and function. DNA is going to be responsible for storing all of our genetic information. It's the instructions that make us who we are. On its simplest level, DNA looks like a very long, thin threads. If you were to unwind and stretch out every single chromosome of DNA in your cell, it would exceed 5 feet in length, and all of that is packed inside of your tiny little nucleuses. So, DNA, short for deoxyribonucleic acid, is going to be a double-stranded molecule that resembles a ladder that is very gently twisted to form a spiral. We refer to this structure as a double helix. Here's an example of what that looks like. So if you were to untwist it and unwrap it, it would look like a ladder. But when you twist it all up, honestly, I think it looks kind of like a spiral staircase. And that's really just sort of what it is. It just slowly, gently twists and wraps around. Each side and half rung is going to be made up from a string of repeating nucleotides. So what we have here is going to be one side of the ladder. These are the rungs of the ladder. Half of that, so if you were to split these rungs right down the center, you're going to see that what you have is a repeating sugar phosphate backbone. It goes phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, all the way down. And then branching off to the side of that, you have our nucleic acids, adenine, guanine, thymine, and cytosine, all the way down. So the section of this, so the phosphate sugar nucleic acid section, or this is what's referred to as a nucleotide. Nucleotides are relatively simple in structure. They contain a sugar. In this case, the sugar is going to be deoxyribose. Does anybody know? I'm going to pause for just a moment. Does anybody know what sugar is contained in RNA? Ribose. In the case of DNA, it's deoxyribose. In the case of RNA, it's going to be ribose. RNA is short for ribonucleic acid. What we're going to have here is one phosphate group in a nitrogenous base. The nitrogenous base just means that it contains a nitrogen. In eukaryotic cells, the DNA is going to be located inside of the nucleus. A very small amount of DNA is going to also be found inside the mitochondria. The mitochondria is sort of a self-sustaining little organelle. It can plug and chug along as it goes. There are going to be 3 main functions of our DNA. It's going to replicate, so that way it can be passed down to the next generation. It's going to store information, and it's also going to undergo change or mutations to help provide genetic variability. So like I said, DNA is a double helix. It's composed of 2 strands that sort of spiral about each other. The nucleotides are made up of those 3 subunits. A pentose sugar, in the case of deoxyribonucleic acid, it is deoxyribose. So there's going to be a pentose sugar, a 5-pointed sugar, and a nitrogen-containing phosphate group, and a nitrogen-containing base. The nitrogen-containing base would be either adenine, guanine, cytosine, or thymine. Adenine and guanine are considered purines. They're made up out of these 2-ring structures. So here's our guanine, it's a 2-ring structure, and our adenine is also a 2-ring structure. Cytosine and thymines are pyrimidines. They only have 1 ring. The phosphate and sugar molecules make up the backbone. So that's going to be this phosphate sugar backbone of the ladder, and then the nitrogenous bases are going to kind of sprint off the side. Or they're going to project off to one side. The bases are going to be joined together using hydrogen bonds, following those complementary base pairs rules. So adenine is always going to bind with thymine. Cytosine is always going to bind to guanine. They bond to each other using those hydrogen bonding. Does anybody remember what a hydrogen bond is? Well, that'd be a great question for our live discussion. So, take a moment to review your notes. See if you can review what is a hydrogen bond, what forms it, and how strong is a hydrogen bond. Is it relatively strong, relatively weak? Where are some other cases in which we can see hydrogen bonds? The two strands of DNA are going to be anti-parallel, meaning they run in opposite directions. In this case, this side runs phosphate, sugar, phosphate, sugar, phosphate, sugar, starting with so we refer to this end that starts with a phosphate, we've referred to this as the 5' end. We refer to this other side as the 3' end. So one end runs 5' to 3', the other end is going to run in the opposite direction. Sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. This is going to run from the 3' end to the 5' end. As you'll see here, all of our bases are also going to be flipped upside down in order to accommodate that the base pairing. So when you're looking at a double helix, one side will have a 5' carbon at one end, the other will have the 3' carbon at the other end. So we refer to this as the 5' end and the 3' end. They're going to run anti-parallel to each other. The phosphate and the sugar molecules that make up the backbone, with those bases that project off to the side, like I said, we're going to follow the complementary base pairs ruling. Adenine is always going to bind to thymine, guanine is going to bind to cytosine. In the process of replicating that DNA, or since DNA is the basic instruction manual for our lives, it's very important that we keep that information intact. Not only do we undergo special precautions to protect it during mitosis and meiosis, but we also want to make sure that when we are preparing to replicate the DNA, we are making an exact copy. DNA replication or DNA synthesis is going to take place before mitotic and meiotic divisions. We want to make sure that we are doing everything we can to make sure that DNA is going to be an exact copy with no mistakes, no mutations happening in the copying process. So we're going to do this through a process known as semi-conservative replication. Semi-conservative replication means that each original strand is going to serve as a template for the formation of a new complementary strand. So what's going to happen is we're going to unwind or separate the two strands of the parental DNA. Here's our original strand of DNA. Here's our original molecule of DNA. What happens is we are going to untwist that ladder and we're going to separate the two sides of the rung. This is our unwinding or separation of the two parental strands of DNA molecule. This is going to be done with a very specific enzyme known as DNA helicase. DNA helicase is going to be responsible for unwinding the DNA helix. Then what we see is we're going to see small little special single strand binding proteins that are going to attach to the newly separated DNA and prevent that helix from twisting back up again. Then we're going to use our complementary base pairing rules to build a new strand of DNA using the template of the old one. So what you see here is we've exposed, when we unwound this, we exposed a sequence that goes C, A, C, G, G. What's going to happen is that free-floating nucleotides are going to come and fill in. They're going to match up, they're going to pair up. So G always bonds to C, guanine always binds to cytosine. So if we have an exposed guanine, cytosine is going to come and fill in that place. If we have an exposed guanine, cytosine is going to come and fill in that spot. If we have an exposed adenine, who is going to come and fill in that spot? Thymine. If we have an exposed cytosine, who is going to come and fill in this spot? Guanine. And here we have another exposed adenine. So a thymine is going to come and bind in that spot as well. So complementary base pairing between the new nucleotides and the nucleotides in the template strand is going to occur. And we're going to fill in all of those missing pieces. This is going to be done with DNA primase. DNA primase is going to place short primers on the strands to be replicated, and then an enzyme called polymerase is going to recognize, it's going to begin sort of that DNA synthesis. The two strands are going to be replicated a little bit differently. DNA polymerase really only works in one direction. So it's going to work by moving in the 3' to 5' direction. So what happens is the DNA polymerase is going to sit on this little strand and he's going to zip along and he's going to help stick those pieces in. DNA polymerase is an enzyme. So just like we talked about earlier with our enzyme analogy, how you can put all those Lego pieces in a box and then and you can shake and shake and shake, but those pieces aren't really going to find each other. A much faster way to do it is to like physically stick those pieces together. That's what's going to be happening here. DNA polymerase is going to sit right here on the template strand. These nucleotide pieces are going to come and DNA polymerase is going to grab them, stick them together, grab them, stick them together, grab them, stick them together. So that's going to be the process, the role of DNA polymerase. Once DNA polymerase has attached, it has performed that complementary base pairing, DNA ligase is going to seal up those pieces. It's kind of like if you were creating a permanent Lego display that you wanted to show off, you would super glue those pieces together. You can do the same thing. DNA ligase is going to super glue those pieces into position once DNA polymerase has put them together. So here is just a series of all of some of the terms that we just used and what their role is going to be. So like I said, the very first step of unwinding the strands of DNA is going to be done by the DNA helicase. The heli referring to the double helix, and the ACE, ASE. Anytime you see that ASE, that typically refers to enzyme activity. DNA helicase is an enzyme that unwinds the DNA helix. DNA polymerase is an enzyme, that ASE is an enzyme that polymerizes DNA. It takes single nucleotides and it turns them into a DNA polymer. DNA ligase is going to be an enzyme that seals DNA pieces together. So that ASE is typically used to denote enzyme activity. DNA helicase is going to zip along the strand of DNA, breaking the hydrogen bonds that are holding the rungs of the ladder together. As it unzips those nucleotides, it's going to unwind the DNA helix, causing the two strands of the double helix to separate and unwind. Once unwound and exposed, the complementary base pairing allows free nucleotides floating in solution to kind of pair up with the nucleotides that are in the now single-stranded DNA. Once paired up with the free nucleotides, they need to be covalently bonded together. Another enzyme is going to be required to do that. That would be DNA polymerase. DNA polymerase is going to be responsible for fusing and creating those covalent bonds. So DNA polymerase is going to link the sugars and the phosphates of the newly attached nucleotides to form the new strand. And then as the double strand of DNA forms, the helix is going to twist around itself again. This type of replication, like I already said, is known as semi-conservative replication. It just means that the original strand splits and serves as a template for the new strand. So in the new daughter strand, one strand belongs to the original parent and the other strand is the newly synthesized strand. The nice thing about complementary base pairing is that it creates two new DNA molecules that are identical to the parent molecule. And the genetic information can then be passed on accurately from parent to daughter cells. DNA replication is a really highly accurate process. The DNA polymerase makes a mistake only about once in every 100,000 base pairs. However, it has the ability to proofread as it goes, and it can correct mistakes that are made. So the resulting accuracy of DNA polymerase is only about one error in every million base pairs. It is a highly, highly accurate process. All right, so the one last thing that I want to mention is that DNA polymerase only works in one direction. It only works from the 3' to the 5' end. So that works great for synthesizing one strand, synthesizing the strand that runs 3' to 5'. However, as we mentioned earlier, these two strands run anti-parallel to each other. One runs 3' to 5', the other runs 5' to 3'. So what's going to happen is DNA polymerase is going to attach and then synthesize backwards and then release. Attach, synthesize backwards, release. Attach, synthesize backwards, and release. These small little fragments are what are known as Okazaki fragments. This is how the DNA polymerase is able to synthesize from the 3' to the 5' direction, whether it is doing the 3' to 5' strand or the 5' to 3' strand. I strongly suggest that for your extra credit videos, you send me a link to a video showing how Okazaki fragments are formed. how they are sealed. This is a great place for you to send me some extra credit videos. All right, we will pick up with the structure and function of RNA in our next video.